Welcome to the Deep Dive. Today we're tackling a big one, a real cornerstone, organic chemistry.
It really is foundational stuff.
We got a chapter here from chemistry, the molecular nature of matter and change. And our goal, really, is to give you, our listener, a solid shortcut through this dense material. We want to make these concepts click, without needing diagrams or models. Okay, let's unpack this.
Sounds good. And it's so important because, well, organic compounds are just everywhere. I mean, think about medicines, food, clothes, fuel. It's all organic chemistry.
Right, it touches everything.
And for ages, people thought there was some kind of mystical vital force needed to make these things. Like you couldn't make them in a lab.
The old vitalism idea.
Exactly. But then boom, 1828, Friedrich Wöhler synthesizes urea, totally organic from inorganic stuff. Game changer. It showed the same rules apply, living or not.
And the absolute key player in all of this complexity and, well, sheer variety is carbon. It really is like the master builder element.
It absolutely is. Carbon's atomic properties are perfectly suited for it. It has 4 valence electrons, right? But forming a +4 or -4 ion, that takes way too much energy. It's just not going to happen under normal conditions.
So it doesn't grab or ditch electrons easily.
Nope. And its electronegativity, it's kind of intermediate, right in the middle, really. Perfect for sharing electrons forming covalent bonds. Strong ones.
Okay, so it likes to share. What about its ability to link up with itself? Those long chains we always see.
Ah, yes, catenation. That's its superpower, really. Carbon bonding to other carbons extensively. Because it's small, those carbon bonds are super strong and short.
Right.
Then you add in orbital hybridization. Remember, sp hybridization.
Vaguely, it allows for different shapes, right?
Exactly. It lets carbon form 4 bonds, pointing in different directions. Tetrahedral for single bonds, trigonal planar for doubles, linear for triples. This gives us chains, rings, branches.
And you mentioned multiple bonds, doubles and triples. They add another layer.
They do. They restrict rotation around the bond. So parts of the molecule get sort of locked in place spatially, adds a whole other dimension to the structures possible.
So what does all this mean for stability? We know carbon compounds can last a long time.
Compared to what? Well, let's look down the group. At silicon, silicon also forms 4 bonds.
Right, group 4A neighbors.
But silicon bonds, much weaker than carbon-carbon, about 226 kilojoules per mole versus 347 for carbon. That's a big difference.
Okay, weaker bonds. But is that the whole story?
Not quite. It's also about the relative bond strengths. For carbon CCl bonds, they're all sort of in the same ballpark, energy-wise.
Okay.
So when carbon reacts, swapping one bond type for another doesn't release a massive amount of energy. It favors stability.
Makes sense. And silicon.
Totally different picture. Cyclo and cycle bonds are way stronger than Cy-Cy bonds. So if a silicon chain meets oxygen or chlorine, reacting is energetically very favorable. Huge energy release.
So it wants to react.
Desperately. That's why something simple like ethane, CH0, is stable in water, stable in air. But disilane, CH, breaks down in water, poof, ignites in air.
Wow. Any other reasons silicon's so reactive?
Yeah, its size. Silicon has these accessible, low-energy orbitals that carbon just doesn't have. These orbitals can basically invite reactants in, making it easier to attack the silicon chain. Carbon lacks that vulnerability.
Okay, so carbon builds strong, stable structures. Now here's where it gets really interesting. The sheer chemical diversity, millions of compounds. How does that happen?
It really boils down to 3 things, all connected. First, carbon bonds readily to other elements. We call them heteroatoms. Nitrogen, oxygen, sulfur, phosphorus, halogens.
So it's not just CC and CH.
Not at all. And get this. Just 4 carbons, 1 oxygen, and some hydrogens. You can make 23 different stable molecules out of just those atoms.
23 from that little handful. That's wild.
It shows the possibilities. Second factor, these heteroatoms introduce differences in electron density. This is key for reactivity. Well, CC and CH bonds are pretty nonpolar, quite unreactive, stable. But a CO bond, oxygen is much more electronegative. It pulls electron density towards itself.
Creating sort of charged ends.
Exactly. A slightly positive carbon, slightly negative oxygen. That polarity makes it a reactive site. Or even bonds like CBr or CS. The polarity difference might be smaller, but the bonds are often longer and weaker, making them easier to break, more reactive.
So these become the action spotters on the molecule.
You got it. And that leads perfectly to the third factor, functional groups. These are specific arrangements of atoms, usually involving these heteroatoms or multiple bonds. Think of them as the molecule's chemical personality.
Like little reactive units.
Precisely. An alcohol group, TASHIF, a carboxylic acid group, COH, an amine group, -amine, each one behaves in a predictable way, no matter what the rest of the molecule, the R group, looks like. They dictate the chemistry.
Okay, so we've got the carbon skeleton, the hydrogen skin, and these functional groups acting as the chemical hands. Let's start simple. Just the skeleton and skin. Hydrocarbons.
Right, compounds with only carbon and hydrogen, the simplest organic molecules, but even here the variety is huge.
How so? Just C and H seems limited.
You'd think, but think about arrangements. 1, 2, or 3 carbons. Only one way to connect them. 4 carbons. Suddenly, 2 ways a straight chain or a branched one get up to 20 carbons. Over 300,000 possible arrangements.
Oh. Okay.
And that's just single bonds. Add 1 double bond to a 5-carbon chain. You get 5 more structures. Make it a ring. Another 5. The possibilities just explode.
And visualizing these, single bonds can rotate freely, right? So a zigzag chain is the same as drawing it straight.
Generally, yes. Rotation is free around single bonds, but double bonds. No rotation. They lock the geometry. That becomes really important later.
Okay, so what are the main types of hydrocarbons?
We usually break them down into 4 classes. First up, alkanes.
The basics.
The absolute basics. They're saturated, meaning only single CC bonds. Every carbon is typically spot hybridized, tetrahedral shape around it. Think methane, ethane, propane, butane, natural gas, gasoline components, waxes.
And they're pretty unreactive.
Largely, yes. Very stable. Their physical properties depend mostly on size. They're nonpolar, so only weak dispersion forces hold them together. Longer chain means more surface contact, stronger forces, higher boiling point.
Makes sense. More sticky surface area.
Pretty much. They mix with other nonpolar things, like oil, but not water. And don't forget cycloalkanes' rings of carbon atoms, like cyclohexane, which famously adopts a chair shape, not flat, to relieve strain.
What's fascinating here is how just slightly rearranging the same atoms can make totally different molecules.
Isomerism, right?
Exactly. Same formula, different structure, different properties. The simplest are constitutional isomers, atoms connected in a different order.
Like the 4-carbon example, butane versus
2-methylpropane, often called isobutane. Same C-Urachori formula. But butane is a straight chain. Isobutane is branched, different shape, different boiling point. Isobutane is more compact, almost spherical.
Ah, so less surface area for those dispersion forces. Precisely. Less contact, weaker forces, lower boiling point. Same thing with COOSO. You have pentane, straight, 2-methylbutane, 1 branch, and 2-dimethylpropane, highly branched, almost spherical. Boiling points drop as branching increases. Shape matters.
Okay, that's connectivity. What about different spatial arrangements?
Now you're talking stereoisomers. Same connectivity, different 3D orientation. A huge area is optical isomerism, involving molecules that are mirror images but can't be superimposed, like your hand.
Left and right hand analogy. Okay.
These are called enantiomers. They arise when a molecule is chiral, meaning it lacks internal symmetry. Often this is because it has a chiral center, usually a carbon atom bonded to 4 different groups.
Got an example?
Sure, the amino acid alanine. The central carbon is bonded to an H, a methyl group, an amino group, NH, and a carboxyl group, COH. 4 different things. So alanine exists as 2 enantiomers, mirror images.
And how do they differ in properties?
Mostly they don't. Same boiling point, melting point, solubility. The key difference is how they interact with plain polarized light. One enantiomer rotates the light clockwise, dextrorotatory, D or plus. The other rotates it counterclockwise, levertatory, L or breg. They're optically active.
Is this just a lab curiosity?
Oh, absolutely not. It's critically important in biology. Enzymes, the catalysts in our bodies, are chiral themselves. They often only recognize and interact with one specific enantiomer of a molecule.
Like a lock and key, but with chirality.
Exactly. Your body uses D-glucose for energy, but it can't really use L-glucose. It needs amino acids to build proteins, not D-amino acids.
Wow.
And sometimes the consequences are devastating. Remember thalidomide?
Vaguely, it caused birth defects.
Yes. It was sold as a mix of enantiomers. One enantiomer was an effective sedative, treated morning sickness. The other enantiomer caused horrific limb deformities in developing fetuses. A tragic lesson in the importance of chirality in medicine.
Incredibly sobering. Okay, moving on from saturated alkenes.
Alkanes are unsaturated. They have at least 1 carbon-carbon double bond. Formula is typically CNH arrow. The carbons in the double bond are spatial hybridized trigonal planar geometry. Think flat.
And that double bond restricts rotation, you said?
Yes. And that leads to another type of stereoisomerism, geometric isomerism, or cis-trans isomerism.
Okay, break that down.
If you have groups attached to the double-bonded carbons, their position relative to the double bond matters. If similar groups are on the same side, it's the cis isomer. If they're on opposite sides, it's the trans isomer.
Okay.
Cis-2-butene versus trans-2-butene. Same CH4 formula, same connections. But in cis, the two methyl groups are on the same side. In trans, they're diagonal. Different shapes, different polarities, different boiling points. They're distinct compounds.
And this cis-trans thing shows up in biology too, right? Something
Okay.
about vision.
Absolutely fundamental division. There's a molecule called retinol in your eyes. In the dark, it exists mainly as 11-cis retinol. It has a bend in it due to a cis double bond. When a photon of light hits it, that light energy is just right to break the pi part of that double bond briefly, allowing rotation. The molecule snaps into the all-trans retinol form, which is straight.
Wow, light flips a molecular switch.
Instantly. That shape change from bent cis to straight trans triggers a whole cascade of nerve signals to your brain. That's literally the first step in seeing amazing efficiency.
That is amazing. Okay, quickly, what about alkynes?
Alkynes have at least one carbon-carbon triple bond, CNH heroes. The carbons are hybridized, so the geometry around the triple bond is linear, a straight line. Like alkenes, they're unsaturated and reactive.
And the last hydrocarbon group, aromatics.
Yeah, these are special. Think benzene, CH is the classic example. They're typically planar rings with delocalized pi electrons moving around the ring. This delocalization gives them extra stability, resonance stabilization.
So they're stable like alkenes.
Stable in a different way. Unlike alkenes that love addition reactions, breaking the double bond, aromatics tend to undergo substitution reactions, swapping an atom on the ring without breaking the stable aromatic system. Some are unfortunately carcinogenic, which is a concern.
Before we leave structure,
Yeah.
how do chemists actually know what they've made? You can't just look at it.
Great question. We have powerful tools. One of the most important is nuclear magnetic resonance spectroscopy, NMR. It's like getting a detailed map of the molecule by probing the environment of the hydrogen atoms or carbon atoms. It gives a unique fingerprint for each compound. Indispensable.
Okay, structure down. Now, the chemical choreography.
Yeah.
What happens when these molecules react?
Right, let's talk reactions. We can group many organic reactions into three main types based on what happens to the bonds connected to carbon. First, addition reactions.
We mentioned those with alkenes.
Exactly. An unsaturated molecule becomes saturated. A pi bond breaks, and two new sigma bonds form. Something adds across the multiple bond, like ethene plus HCl gives chloroethane. These are often exothermic. Remember the bromine test. Orange bromine adds across a CC bond, and the color disappears. That's an addition reaction.
Got it. Add things, lose the double bond. What's the opposite?
Elimination reactions. Here, you start saturated and become unsaturated. You form a multiple bond by eliminating atoms or groups from adjacent carbons. Often, a small, stable molecule like water or HCl is removed. It's the reverse of addition. Think of removing water from an alcohol to make an alkane.
Okay, addition makes bonds, elimination breaks them to form double, triple bonds. What's the third type?
Substitution reactions. Here, one atom or group attached to a carbon is replaced by another. The total number of bonds to that carbon doesn't change. It's a swap. Like making banana oil and ester, where an oxygen group replaces a chlorine atom on a carbon chain.
Add, eliminate, substitute. Seems straightforward. What about redox, oxidation, and reduction?
Good point. Organic chemists often think about redox in terms of electron density around carbon. Oxidation generally means carbon forms more bonds to oxygen, or other electronegative atoms, or fewer bonds to hydrogen. Basically, carbon loses electron density.
Like burning fuel.
Yeah.
Combustion.
That's the most extreme oxidation, yes. Burning ethane to co-uros in water. A more controlled example is oxidizing an alcohol, say 2-propanol to a ketone, 2-propanone. The carbon bonded to oxygen gains another bond to oxygen in the CO and loses its bond to hydrogen.
Okay, and reduction.
The reverse. Carbon gains electron density. It forms fewer bonds to oxygen or more bonds to hydrogen. A classic example is adding hydrogen H0 across an alkane's double bond to make an alkane. That's a reduction of the carbons involved.
So these functional groups we talked about really are the reactive personalities driving these reactions based on their electron distribution.
Absolutely. Their polarity, their bond types that dictates whether they're likely to undergo addition, elimination, substitution, oxidation, or reduction. Let's hit a few key ones. Alcohols, ROH. That OH group allows hydrogen bonding, so smaller alcohols mix well with water, have higher boiling points than similar-sized alkanes. They can be dehydrated, elimination to alkenes, oxidized, as we said, to aldehydes or ketones, or undergo substitution. Versatile.
Then aldehydes and ketones?
Aldehydes are CHO, and ketones both have the carbonyl group, COO. It's very polar, electron-poor carbon, electron-rich oxygen. Prime site for addition reactions. You can reduce them back to alcohols. And really cool, you can use organometallic reagents like Grignard's RMGX to add new carbon chains to that carbonyl carbon, building bigger molecules.
Building carbon skeletons, nice.
Electrolyte acids.
Carboxylic acids are COOH. They have the carboxyl group. They are weak acids, donating a proton in water. Think vinegar. Acetic acid. They react fully with strong bases. Fatty acids, crucial in fats and oils, are just long-chain carboxylic acids.
So we've gone from simple molecules and reactions to these functional groups. But what about the really giant molecules, the plastics, the proteins, polymers? How do we get there? The monomer-polymer theme. It's central. Polymers are huge molecules made by linking lots of small repeating units, the monomers, together. Nature does it, and we do it synthetically.
Okay, synthetic ones first. Plastics and stuff.
Right. Two main types. Addition polymers. Unsaturated monomers, usually with CCC bonds, just add to each other in a long-chain reaction, like ethene monomers linking up head to tail to make polyethylene plastic bags, bottles. Teflon, polypropylene are others. We can control the catalysts now to fine-tune properties like density, flexibility.
Adding up the monomers, what's the other type?
Condensation polymers. Here, monomers usually have two functional groups. They link up by eliminating a small molecule, typically water, between them. Think polyamides, like nylon. You react a molecule with two amine groups with one with two carboxylic acid groups, water splits out, and you form strong, flexible amamide links. Or polyesters, like Dacron PET, react a diacid with a dialcohol, eliminate water, get ester links, used for fabrics, films, bottles.
So addition just adds up, condensation kicks out water.
Yeah.
Humans got pretty good at this, but you said nature did it first?
Oh, way first. Biological macromolecules are nature's polymers, built using the same organic chemistry principles. Masterpieces, really.
Like?
Sugars and polysaccharides. Simple sugars, monosaccharides like glucose, are the monomers. They link via condensation reactions to form disaccharides like sucrose, table sugar, or huge polysaccharides. Starch is how plants store energy. Cellulose gives them structure. Both are just polymers of glucose.
Wait, if they're both glucose polymers, why can we digest starch but not cellulose?
Different linkages. The way the glucose units are connected, the orientation of the bond between them is slightly different. Our enzymes recognize the starch linkage, but not the cellulose one. Structure dictates function.
Okay, what else does nature build?
Amino acids and proteins. Proteins are polymers of amino acids. Each amino acid has an amine group, a carboxylic acid group, and a unique side chain, all attached to a central carbon.
And they link up how?
Condensation reaction again. The carboxyl group of one reacts with the amine group of the next, splitting out water and forming an amacid bond, which in proteins we call a peptide bond.
So a chain of amino acids, that's a protein.
Or it's the primary structure, just the sequence. But then that chain folds. Hydrogen bonding along the backbone creates localized shapes like alpha helices and beta-pleated sheets, that's secondary structure. Then the whole thing folds into a complex 3D shape based on interactions between the R groups, that's tertiary structure. And sometimes multiple folded chains, subunits, assemble together a quaternary structure like in hemoglobin.
And all that folding is just dictated by the original amino acid sequence.
Fundamentally, yes. The sequence determines the shape, and the shape determines the function. Amazing. Enzymes, antibodies, hormones, all proteins with specific shapes for specific jobs.
Mind-blowing complexity from simple building blocks.
Oh.
Okay, the ultimate biological polymer.
Has to be nucleic acids, DNA, and RNA, the blueprints of life. The monomers on nucleotides. Each nucleotide has a sugar, deoxyribose in DNA, ribose in RNA, a phosphate group, and a nitrogen-containing base, A, G, C, T in DNA. U replaces T in RNA.
And they link up.
Phosphate of one links to the sugar of the next, forming a long sugar-phosphate backbone with the bases sticking off. Condensation again, forming phosphodiester bonds.
And DNA is the double helix.
The famous double helix. Two strands coiled around each other. The backbones are on the outside, bases pointing inwards. And the key is specific base pairing via hydrogen bonds. A always pairs with T, G always pairs with C. This holds the strands together and keeps the helix diameter constant.
And this sequence of bases, That's the code.
That's the genetic code. It dictates everything. In protein synthesis, the DNA sequence is first transcribed into messenger RNA, mRNA. Then the mRNA code is translated at the ribosome into the specific amino acid sequence of a protein. RNA makes protein the central dogma, simplified.
And how does it copy itself?
DNA replication. The helix unzips, and each strand acts as a template to build a new complementary strand using those base pairing rules. Ensures the genetic information is passed on accurately.
And understanding all this has led to, well, huge technologies.
Absolutely. DNA sequencing lets us read the Base Order Think Human Genome Project. And DNA fingerprinting, looking at unique repeating sequences, is invaluable in forensics and identification. It all stems from understanding this molecular structure in chemistry.
What an incredible journey. We started with just carbon, this amazing versatile builder, saw how its simple properties lead to immense structural complexity, isomerism with huge real-world consequences like with thalidomide or fission. Then we moved through the basic reaction types, saw how functional groups give molecules their personality, and finally scaled up to the polymers, both synthetic and the biological ones that literally define life. Polysaccharides, proteins, DNA, it all connects.
It really does. It's, well, elegant, isn't it? How a few fundamental principles about bonding and structure cascade up to create the complexity of life and materials science. The main takeaway, I think, is that understanding these building blocks, carbon's abilities, functional groups, reaction types, changes how you see the world. You start to recognize the molecular patterns everywhere.
It definitely gives you a new lens. We really hope this deep dive has helped make the world of organic chemistry a bit less intimidating and maybe even exciting. Keep exploring, keep asking questions. From all of us here at the Deep Dives, thanks for tuning in.